successful for two main reasons. People, good people. Not the smartest people, but the best people for the job. Some of them were smart. They all worked hard, all incentivized to do a great job, and some good basic technology. Technology which had legs on it, which we could build on and go into different areas with. And that's basically it. The products, there are really two classes of products. One, diagnostics, to detect diseases so that they can be treated early. And the two biggest diseases are chlamydia and gonorrhea, two sexually transmitted diseases. And if chlamydia, for example, which is usually asymptomatic in females, isn't treated, it can lead to pelvic inflammatory disease and infertility. And if you detect it, you can treat it with a short course of generic antibiotics. So that's the sort of value we bring to the equation. And the other part of the business, and those two combined for about 85% of revenues, is blood screening. Blood screening is where a person will donate blood, and before it's transfused, they'll use our tests to make sure that it's free of HIV, hepatitis B, hepatitis C, and West Nile virus. Good business. And we'll explain a little bit more about that. So basically, GenProbe makes tests, uses DNA probes, put it into a kit. The kit is shipped to a hospital lab or a clinical reference lab. The physician takes a sample, sends it to either a hospital lab or a clinical reference lab. They use our test, and increasingly, they're using it on our instruments. And that's a picture of one of our instruments, the first in class, really, the first high volume, totally automated, standalone system. The first that's ever been done by a little company in San Diego competing with Roche, Abbott, Becton Dickinson, Biomedicina, and others to come up with that, because we had good people and good technology, a lot of confidence. This is the size of the molecular market, and I'll explain what molecular means a little better. Basically, we take a little piece, and of course it's not even visible in front of me, a little piece of DNA, and use that to look for its counterpart in specimens, in urine or in blood or in swabs. And if it binds to that, a light will go off, and it's either a yes-no decision or we can quantitate it, saying there's a lot of this in there or there's just a little bit in there. And that'll become clearer later. So these are the parts of the business. The first, the 430 million CT and GC, that stands for chlamydia and gonorrhea. HPV, that's the human papillomavirus. That is the cause of cervical cancer. We are about to file for our approval of that product in the US. It'll be done by the end of the year. The product has been approved in Europe for about 3.5 years and is doing really well. Blood screening, we've talked about. Virology, basically it's cold, flu viruses. HAI, that's the maroon one, that is hospital-acquired infections. That would be, and I don't know if you know these or not, but one of the reasons to get out of a hospital quick is because people can get pretty sick in hospitals. They can pick up an infection. MRSA is methicillin-resistant Staph aureus. It's resistant to most antibiotics. If you get it, you may never leave the hospital. Vancomycin-resistant enterococcus, VRE, is another one. And there's a third that is just starting in North America. It's been in Europe for a while, C. difficile. And they all have to do with hygiene. You know, they're fairly simple to stamp out, but some hospitals have a 40% infectivity rate. Now, you can go online and find out what the infectivity rates of these hospitals are and stay away from them. There's nothing worse than going into a hospital sick and either not coming out or coming out with a bigger infection and problem than you went in with. So it's a big market, over $4 billion and growing nicely. A little history of GenProbe. I won't bore you with that. The importance of patents. GenProbe has over 530 issued patents. Some pretty productive R&D people, and they're all used. And they give GenProbe a tremendous advantage versus competition. Now this is neat, and I hope it works, because what you're seeing there is a DNA probe. Now let me give you a little biology lesson. And again, this is from a person who's never taken chemistry or biology. I went to a Jesuit school in Baltimore. We're going to run that again, if you can hear me when I finish talking. And they decided that it's better to study 2 years of Greek than to take biology and chemistry, not knowing that I'd spend 40 years in biotech and healthcare. Every organism has a stretch of its DNA that is unique to that organism. And if we don't know what that is, we can sequence it and find out by comparing it to other genomes. There are only 4 chemicals that make up DNA, and DNA is a recipe for life. We'll call them A, T, C, and G. A will only bind to T, and C will only bind to G. And it's that complementarity that allows us to identify organisms. So what you're seeing there is a DNA probe. Looks like it's made out of rubber. It's bouncing around a lot. But attached to the DNA probe is a source of light. And when it binds to the exact complement of that, the light will go off, and you get a positive. So if, for example, we're looking for tuberculosis, we'll take a throat swab, doctor will, send it to a lab. The swab goes into a little tube. That tube contains, this is the GenProbe tube, the ingredients to break apart the cell and to free up the sequences of the bacterium. We then put our complementary probe in there after amplifying it, and if it finds it, a light goes off and you get a positive. Could you run that last one again? If you can, I'll understand. But that's the essence of a DNA probe. Again, a short stretch of DNA that is complementary to the unique sequence of the bug we're looking for, be it tuberculosis, HIV. Thank you very much. There we go. So what you're seeing are probes, the green part, and the stretch, the organism, the white part. And it'll go along, and it'll come up because what we've done is taken a stepladder apart. A is one half of the rung, T is the other half of the rung, and it wants to get back together again. So there's a certain affinity between the probe and the sequence of the organism we're looking for. And it is an extremely precise match. And that could be the sequence A, T, T, G, C, 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 and run on for 40 or 50. So if that light goes off, you know you've got that disease. We talk about sensitivity and specificity in diagnostics. And the specificity means that you're going to detect only the organism you're looking for, not another one. And that's the problem with monoclonal antibodies. And that will give you a false positive. It's not there, but you think you found it by attaching to something. That's not good. The other thing is sensitivity, is the ability to find the organism even when it's there in a very, very small quantity. And if it's there and you don't find it, that's a false negative. And that's not good because you want to treat it, you have the disease, and you don't think you have it. And the sensitivity and specificity on those probes that GenProbe has, 99.7% and 99.8%. So terrific, and much better than competition. I'm going to zoom through the rest of this, if you let me. This was in a separate file. This is an example of clinical utility, and every product has to be better than the product that was before it. In this case, the National Heart, Lung, and Blood put out a contract to use molecular diagnostics, molecular technology, to look for HIV, hepatitis C, hepatitis B in the blood supply, because they were concerned that the antibody tests weren't finding it soon enough. And this is exactly what happened. So today, I get infected with hepatitis C, as an example. The virus replicates, it grows. After about 2 weeks, it reaches a certain level, but continues to grow. The immune system doesn't notice this until about 30 or 35 days after infection. The immune system then generates antibodies and this is what we talked about before, the antibodies will bind to the virus as a big flag, and then the macrophages will come, if it's not too much, because there are only a limited number of macrophages, and eliminate the threat. Well, the competitive test to a DNA probe is an antibody test. And the antibody test, immunoassay it's called, detects the antibodies. Well, the antibodies will only be detected 60 days after infection. Positive EIA test. But people are receiving that blood before the immunoassays can detect it. And if they get the blood anytime when the virus starts to replicate, they're going to have hepatitis C. So they asked GenProbe to develop a nucleic acid test, an NAT test. And in the first try, we got it after 28 days. Now it depends on the virus. Sometimes we've gotten it after 3 days. So the window it takes to detect it after you have the virus has been 60 days. It's been closed now to about 10 days, effectively. So a lot of people were getting blood that was tested with the antibodies at, say, 45 days after infection. They said, it's okay. It wasn't okay. And the GenProbe test can detect it because it's more precise. And it goes after the virus directly, not the antibodies that take a while to react to the virus. The gentleman on the right is the former president of the United States. It was a great honor for me to go to the White House and to accept the National Medal of Technology. It was the 2004 National Medal of Technology, but in all reality, because Washington, I didn't get it until 2006. It took them a while to do all that, to vote on it. But it was a great honor, and GenProbe is the only company in San Diego to have received the National Medal of Technology. It is established by Congress and the companies who embody the spirit of American innovation, who have advanced the nation's global competitiveness. Good company, IBM, Amgen, Biogen, and all the rest of that. And we were honored for the development of that test for hepatitis C. Saved a lot of lives. Nice place to work. If you ever come to San Diego, you're looking for a job, give us a call. We are always looking for great people. Always. Growth, nice growth. A little bit over $500 million is expected this year. Again, that's a forward-looking statement. Might not happen. And earnings per share, pretty good. Nice growth, growing nicely. In 2008, I should say that it looks the same as 2009, and it was, but we had 2 events in 2008 which were one-time, non-repeatable. We received a $10 million milestone payment from Novartis because of a product that was launched and approved. And we also received about $16.4 million from Bayer. We were in a litigation with them, and that was part of the settlement. So if you took those 2 out, because we can't expect them to occur every year, the earnings per share would have been about $1.65. So nice growth from 2007, yet 2009 was nice growth. And these are non-GAAP, I believe. I told you I'd tell you something that was non-GAAP for historical comparative reasons. A lot of money, good balance sheet, generates an awful lot of cash. And the strategies for growth is take all the cash, and there is a limit as to what we can do organically in the bloodstream.